All right, let's move on now to the next chapter that we have, or section of the chapter that we're covering right at the moment. This is chapter one, section two, microbes in history, and this section is informative. It's really designed just to kind of give you an appreciation for what's going on, and this is still part of the week one. And again, we're trying to clump these as in groups of 2 weeks for exams and that sort of thing. And doing it week by week, I think, helps at least conceptually for you to kind of wrap your brain around the 1 week at a time and then putting it all together. I hope the organization of this is helping you out. So what we're looking at is the timeline of development of microbiology from the 1600s, and then some recent discoveries and the impact of these things. And of course, the scientific method made it all happen. So because we couldn't see things, of course, when streams and ponds kind of dried up a little bit, all of a sudden frogs appeared. Spontaneous generation. I mean, they just appeared, you know. And so that was sort of the momentum that was going on. It was a generally accepted idea of spontaneous generation. And this goes back to Leonardo da Vinci days, where he was thinking that little sperm cells had little people in them, little babies, and then the babies would grow once the seeds were planted and that sort of thing. And obviously, we didn't see it. And so the concept really started to come down to the discovery of microbes with the first microscope and looking at Leeuwenhoek's day when he could see little things, little animalcules in the droplets of water and that sort of thing. So the discovery of microbes, a belief of abiogenesis was still embraced. In other words, that there was no real life that begat it; they just spontaneously formed. And the idea actually became biogenesis, saying that all living things arise from others of the same kind, and which today, you know, is obvious, that's true, but it didn't always be like that. Now, I've already mentioned that Louis Pasteur was one of my really, I admire the work of Louis Pasteur, and he studied the roles of microorganisms in fermentation, beer, and wine, and I've mentioned that he kind of capitalized on that concept to fund his research. But one of the things that he did is he put to bed this notion that things that are abiogenic begat life, and the idea was he came up with this open or Swan-Neck flask, which was open but had sort of a dip in it that things couldn't just blow in there and get into it and contaminate it. It's open because everyone said, well, if you close it, then life is snuffed and blah, 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 and can't spontaneously form. So he left it open, and it still sits in the museum today without any life in it, open flask. But the Swan-Neck flask is was a very clever way. So he heated to sterilize the broth and really to kind of sterilize the whole thing. And flasks that were exposed to dust from the air showed microbial growth. Flasks exposed to the air but not to the dust. In other words, with that little swan neck, it sort of acts like a sort of a uh, like in a drain, that little area that slumps, that things can't get above that. And that really was very clever and was able to do that. So you can see that in this drawing here of the swan neck. So if you opened it up, things could get in it just by contaminating. And this is part of the aseptic technique I wanted to at least bring up right now, because things just will randomly contaminate as it's around. And so based on that, then we sterilize that and keep that swan neck. And you can see that little sort of dip in the area right about in here. We see that dust and things would kind of build up here, but it's still open, so he could snuff the naysayers saying, well, you know, but they can get access to the environment. So it's just the dust and things carrying microbes can't get in there, and as a result, nothing would grow in the flask. And that was the Swan-Neck flask experiment, and that put to bed the whole thing. So the role of the microscope, sort of around the same sort of time, studied household objects, plants, and trees, and described cellular structures and drew sketches. Robert Hooke was actually very good with lenses and the concepts behind those. He could actually produce those sorts of things, and that was his gift. And he created a document that documented all of the drawings of what he saw with. He made the drawings of things that he saw, and Microbiome was a publication that he penned that was really important. And so Anton Leeuwenhoek actually was given the idea, or given the title, that he was the father of the microscope, but others were really kind of before him, but he's the one who wrote it down and was famous for it, and Leeuwenhoek got the recognition. So he manufactured a really simple microscope, and he observed these little animalcules in sort of a little drop of water, and then he constructed all sorts of microscopes that deviated on it, but it's really no better than a magnifying glass that you get at, let's say, at Walgreens or something. About 200 to 300 magnification. It's 300x, which is, it's about as much as a drop of oil adds to looking at a slide under the 1000x microscope, and you get an additional 250x with a drop of oil. I'll mention that in the lab and how that happens, but it's about as much as Leeuwenhoek's microscope was in terms of its ability to magnify. So Ferdinand Kohn discovered and described the heat-resistant endospores, which was really important because endospores, all of a sudden, even though you'd heat it, not sterilize, but heat it that they thought that would clean most things, and things would grow, and it came out that there's these spores. Now, we know and heard of the revenge in the Egyptian times where things would grow. Well, you have dead objects or dead people that were buried a long time ago in really nasty, dirty linens. They reused a lot of linens for various things, and then they'd wrap the dead with them. But the thing is that they had spores. And then people would go and into the tombs, and they unraveled and look at all the gold and blah, blah, blah. And what you do is kick up dust, and spores would be in them. They'd germinate in your lungs and wherever. And you would come down with whatever the spore form originally was. It's usually the spore formers can be nasty pathogens, the Clostridium species and things like that. And, of course, in those days, if you developed a cough and all of a sudden you had really serious problems, you probably died from it. It was not a revenge or a curse or anything like that. It was just the fact that you had spores that germinated, and you were dealing with dirty linen. I mean, come on. It's kind of where we are. Oliver Wendell Holmes and Chamelwes described the importance of hand washing and preventing disease. These, really, as obvious as that may seem, it was quite important. And we're told now about hand washing with COVID-19 virus and all that. It's true. It's really important to wash your hands, not touch your face, and that sort of thing. Joseph Lister was a surgeon, and he was really getting upset that he'd do these amazing surgeries and patients would die. So that's where the concept of the operation was a success, but the patient died, and he wanted to deal with that. So he would have up in the ceiling these carbolic acid sprayers, and they would spray. And the idea is that they would kill the bacteria and various things, even viruses that they didn't know about in those days. But the problem is that the surgeon, by the end of the day, would have these serious lesions on their arms and things from the carbolic acid, which, but the patient survived better. Of course, they didn't have the infections, and that's where aseptic technique started to come in during surgeries, and that sort of was the standard, or the in terms of proof that aseptic techniques actually do work. The continuation of Louis Pasteur's work, in addition to the Swan-Neck flask, he invented pasteurization, which was named after him, and milk is pasteurized and various things. And the idea behind that is that you flash heat it and you kill most of things. Now, obviously, that doesn't cover the spores, but you pasteurize it and you get most of it, and that's good. You cut back on the contamination load. And he also was the first to really kind of link human disease to infection. Now, he's the one that kind of termed the word virus. He viewed that as a toxin or toxic material or something, and is Latin for that. But the idea is that he related these things to disease, which was, to us is obvious, but at his time that was really quite amazing to do that. So Robert Koch's postulates a series of logical steps, which viruses, by the way, violate every time, although some will claim that they can prove Koch's postulates for viral infections. It's just hard to regrow the organism like viruses, because you have to have a host to grow it in, and then that kind of wasn't really intended for that. It's for mostly bacteria types of diseases where you could isolate it from an organism, culture it, get it in pure culture, and we're going to talk about how that's done, and then you reintroduce it, and if you can reproduce the disease with the organism that you introduced and then recover that same organism, you've now satisfied Koch's postulates, and that shows that that they're pathogenic and they cause disease, which is still used today. Anthrax was caused by Bacillus anthracis in 1875 using Koch's postulates, which at the time that was really sort of the high bar, high standards in science. And then I've already alluded to, but Daniel Nathans, Werner Arber, and Hamilton Smith discovered a little molecular scissors that bacteria produce. We call these restriction enzymes, and they always cut DNA at a particular place. And you can see that, interestingly, CTAG, if you look at the, flip it around on the other side, it's CTAG, so they're concatenamers. They always seem to, restriction sites are that way. It's kind of interesting. But when they cut them, they always cut them the same way, and then you can use DNA ligase and put them right back together. And that is what the revolution of genetic engineering is really based upon. Polymerase chain reaction, PCR. We've done this in the 110 and 111 labs. And really all PCR does is amplify DNA, but the DNA that it amplifies is very specific. So the segments that you see in that sort of skin colored area, what they call primers, those we can synthesize with a DNA synthesizer and pick out specific regions of DNA that we want to amplify. If we can amplify it, then the DNA regions were there. If you can amplify it, then the DNA was there. If you can, of course, it wasn't there. And that's called. There's a technology that kind of utilizes that concept, and it's a technique that has been used, and it's very popular these days. And we use that to identify really quickly if a microorganism's present, let's say, in a patient that takes, let's say, 4 hours instead of growing it overnight and that sort of thing. And they call it DNA fingerprinting, and it really is a very nice way to do business. And it's very specific. You can detect nanograms, so they're very small amounts, picograms, nanograms, and get on down there. It's a very small amount. And they also use it in forensic science, because you'll leave your DNA behind, usually in a crime scene. It doesn't take much. We can amplify it and then run tests on that. So it's a very, very powerful technique. And again, it takes advantage of knowing the chemical properties of DNA. When you heat it, it separates the two strands, and you cool it a little bit, and you can hybridize a little primer segments and then use a special DNA polymerase from a microorganism that grows in higher temperatures normally, the thermal vents and things. And this thermophile can grow in that. So the enzymes can handle the high heat. So we use the DNA polymerase, we call it Taq polymerase, and we utilize that to amplify the DNA, because we can leave the heat there to separate the DNA, originally cool it a little bit, get the primers to bind, and then the DNA polymerase will synthesize the new strands, as you can see in this diagram here. So it's a very clever technique. And small RNAs, one of the things that we assumed originally was all about DNA, but we're finding out that DNA isn't the key player all the time. It seems like RNA. Now we know that from tRNA, transfer RNAs, I mentioned before. Transfer RNAs are the key, sort of the decoder ring between the sugars of DNA and then the amino acids of proteins, and it acts as the deciphering key between those two systems. There's nothing like it that we know of in living systems other than that kind of decoder ring. But the transfer RNase is a folded RNA molecule that's double-stranded in most of the regions when it folds, and it creates this 3D structure as a result. It's quite amazing. Well, we're finding out that RNA has gotten to be really important, and what we find is that some of these RNA molecules regulate a lot of things, and so the RNA is not to be underestimated. It's very important. And we also know that the RNAs in the bacterial world are very important. But that's about all I'm going to say for that right now. And so utilizing these tools like DNA barcoding and the like, and knowing to identify these. Now, I used to do it the old hard way, using a whole set of biochemical tests to identify certain microorganisms. It would take weeks and weeks of a lot of labor-intensive work. I mean, just awful stuff. I used to do stool samples and things. Wow, it was a lot of work. But the Human Microbiome Project, we've now can utilize these DNA tools that allow us to use barcodes and things in the philosophy of PCR, and to identify the presence or absence of these microorganisms. And we can identify where these live in various regions, and now we understand where these microorganisms hide out, and that's what the heart of the Human Microbiome Project tells us, where and how diverse these microorganisms are. It's quite amazing, and we'll talk more about that at a later date. So that is section 1.2. I'll stop there, but we will be moving forward in the next series of lectures here on the outcomes of looking at the biochemical components. And I'm going to have you refer to a website that you can go to look at these things. I do a lot of the 3D printing at Wake Tech, and one of the things that we can do is 3D print protein molecules and actually put our hands on them and look at them. And so it adds a unique sort of aspect to our understanding of these. So that's what we're going to be talking about, looking at cellular components and the families of biochemicals. Now, in that discussion, of course, anytime you meet a microbiologist, they will bleed as a biochemist. And if you're interested in microbiology, then obviously a lot of your coursework will include biochemistry, chemistry, and various genetics behind that, and it's just kind of how the field is. But anyhow, I will catch you at the next lecture and be posting these in the same sort of general area that you found this one. So thank you for your time, and we'll be chatting with you in future films.